So I think we're going to do questions at the end. And so I'd like to introduce Amy Fuller, one of our other genetic counselors, who's going to be talking about deletion duplication testing. Thank you.
Thank you, Liz. Can you guys hear me in the back? Everything is okay? Okay. So for those of you who are familiar with GeneDx testing, many of our tests, especially the deletion duplication analysis, have the suffix DX. So I'm sure even those of you who are familiar with our services get confused from time to time with all the DXs. It's copy DX, exon array DX, genome DX, fish-on-chip DX, and then the list literally goes on and on. So I'm here today in hopes to shed some light on each different technology and hope to help you decide which DX can lead to a diagnosis. So by the end of this talk today, I hope to outline the different deletion duplication testing methods available today and discuss the advantages and limitations of each different technique. I'll divide the talk today between genome-wide applications and gene-specific techniques and use case examples to highlight their differences. So classic sequence analysis and PCR cannot identify large gene deletions, duplications, or rearrangements. Routine PCR products do not reflect amounts of target in the sample. So in sequencing a patient with a heterozygous deletion, normal sequence data is generated from the non-deleted allele. And due to the lack of quantitation, one copy can look the same as 2 or 3 or even more. So in humans, gene expression typically occurs from both alleles. Loss or gain of an allele can have major effects on development and function. So consideration of large gene deletions is particularly important for disorders that arise due to loss of function of one allele or haploinsufficiency. So today I'm going to start by talking about genome-wide applications of deletion duplication testing. And so I will address first targeted array CGH. Oh, I'm sorry. I actually will first be discussing whole genome array CGH, and then move on to targeted array CGH, which is just a smaller version with oligonucleotide probes as well. And then just briefly, I will touch on SNP genotyping. Okay, so the array CGH techniques, I think pretty much everyone here is familiar, but just as an introduction, it works by applying differently labeled fluorescent dyes to patient and control DNAs and hybridizing them together on a slide customly printed with unique probes and scanned to detect the fluorescent signals on the slide. Okay, and just to show the basic images of the results. So where you see signal intensity dip, that would represent a chromosomal deletion, and when you see the genomic gain or jump, you're looking for a duplication or amplification. So at GeneDx, the whole genome application, we identify it as Genome DX, and it's a 105K oligonucleotide probe design. And the advantages, the clinical usage is for detecting genomic imbalances in individuals with unexplained dysmorphic features, birth defects, mental retardation, developmental delay, seizure, and ASD. And the advantages of this application is that it can detect deletions or duplications that are 50 kb in targeted regions, those that are associated with known contiguous gene deletion or duplication syndromes, and approximately 200 kb in other areas of the genome. It can also help determine boundaries of abnormalities that were identified by routine karyotyping and identify, obviously, novel microdeletion duplication syndromes. And the disadvantages, it obviously cannot identify any balanced rearrangements, and polyploidy, low-level mosaicism of approximately less than 25%, and small DNA mutations such as point mutations or other small mutations identified that should be identified by routine sequence. And so here's just a really good Genome DX case example that came across recently that highlights the utility of the genome-wide applications, as well as a friendly reminder for all of you to please include clinical data on your laboratory requisition form. So this case came to us with a 2-month-old male. There was no clinical information provided on the requisition form. And on our 105K array CGH, we identified a small 139 kb deletion within cytogenetic band 22q13.1. And within that deleted interval, there were 3 genes of unknown function that were reported to vary in copy number in the general population. However, there was one gene upstream of the deleted interval that is clinically significant. It is a SOX10 gene, and it's one of 3 genes involved in Waardenburg syndrome type 4 or Waardenburg-Shaw syndrome. And WS type 4 has symptoms of both classic Waardenburg syndrome as well as Hirschsprung's disease. And so we were questioning whether or not any region of this SOX10 gene was within our deleted interval, because it's just immediately adjacent. And we designed qPCR primers targeted to intron 2, so the very beginning of the gene. And our results showed the presence of 2 copies, indicating the deletion did not include the coding region, this particular coding region of the SOX10 gene. And so here's just an image of the array CGH result, and you can see where the yellow arrow is pointed. I don't have a pointer. I think you can see the yellow arrow. That's okay. It's pointing to the SOX10 gene, and it's immediately flanking the deleted interval. And to further investigate this case, we asked that both parents be submitted, both parental samples be submitted, and we identified this deletion to be de novo. And upon that conversation asking for parental samples, the physician did confirm that this patient was presenting with heterochromia and short segment Hirschsprung's disease. So this immediately confirmed the significance of this deletion. And so just to briefly summarize SOX10, the mutation spectrum, most of them are truncating mutations, nonsense and frame shift mutations. Partial deletions have been identified within the SOX10 gene, and approximately 5% of all Waardenburg syndrome type 4 cases are due to SOX10 deletions. However, we couldn't find anywhere in the literature of deletions outside of the SOX10 gene that were causative. And so this patient's deletion, including the flanking region of SOX10, obviously was preventing normal function of the SOX10 expression. And this is just a great example of the importance of genome-wide applications. This was able to identify an abnormality that may have been missed had the physician ordered a more gene-specific deletion duplication technique.